Welcome back to my classroom, and today we are going to discuss the process of photosynthesis. As we all know, photosynthesis is a process by which carbon dioxide reacts with water in the presence of sunlight and chlorophyll to give you your sugars, water and oxygen as byproducts. But when we look at photosynthesis as a chemical reaction, then what we can see is that photosynthesis is a reduction-oxidation reaction. And if you look at this diagram closely over here, you find that these are the reactants depicted over here, which is water and carbon dioxide, and you have the products which are oxygen, water, and your sugars. And if you break down this reaction, what we can see is that carbon dioxide over here is reduced to give you your sugars, and your water, on the other hand, is oxidized to give you oxygen. So now photosynthesis chemically can be considered as a reduction-oxidation reaction, where water is oxidized and carbon dioxide is reduced to give you your products. Now, photosynthesis takes place in the chloroplast. This is what we all are aware of. So you have chloroplasts which are present in the mesophyll cells and are present on the plant, and it is in these chloroplasts that you have the entire process of photosynthesis taking place. And this process is broken down into two very distinct reactions, where one reaction takes place in the thylakoids of the chloroplast and another reaction takes place in the stroma of the chloroplast. And what we are going to look at in this classroom is we are going to try and understand what really happens in these two very distinct but very separate reactions that you have in photosynthesis. So if you break down photosynthesis, you can actually break down photosynthesis into the light-dependent reaction that takes place in the grana, and also the light-independent reaction which takes place in the stroma. Now, the reaction in the grana requires light. It produces ATP and NADPH, and oxygen is produced as a byproduct. And in the light-independent reaction, this reaction that takes place in the stroma does not require light, but it utilizes the ATP and the NADPH to produce the carbohydrate, and also it is in this reaction that the plant or the chloroplast incorporates carbon dioxide into the process of photosynthesis. So first we will look at how does the plant absorb light. Well, the plant absorbs light by the help of pigment which is called chlorophyll. And you have two very distinct chlorophyll molecules that enable the plant to absorb light and utilize this light for the process of photosynthesis. And you have chlorophyll A and you have your chlorophyll B. And they have a similar structure in which they have a porphyrin ring and they have a hydrocarbon tail, but the only difference is that chlorophyll A has a CH3 group and chlorophyll B has the CHO group in the porphyrin ring. And this difference allows chlorophyll molecules to absorb light differently. So what we find is that when you look at the absorption of light by chlorophyll, your chlorophyll A molecules absorb light in the region of 450 nanometers, which is in this region, and also can absorb light in around the 630 to 650 nanometers. The chlorophyll B, on the other hand, can absorb light at close to 500 nanometers, and also they can absorb light at close to 680 to 700 nanometers. And the presence of these two types of chlorophyll allows the plant to expand the spectrum by which it can absorb light. Apart from these, you also have the carotenoids, and what these carotenoids do is they further enhance the spectrum of light that the plant can absorb and utilize for the process of photosynthesis. So when you look at this, what we call as the absorption spectrum for light in photosynthesis, this is strikingly similar to what we call as the action spectrum of photosynthesis. And what we can see is that wherever you have absorption of light, there you have very high rates of photosynthesis as measured by amount of oxygen released, and where you have low absorption of light, there you tend to have lower rates of photosynthesis. Again, when you look over here, the high rate of absorption of light will also allow the plant to carry out more photosynthesis, and therefore the rate of photosynthesis is also enhanced. So this presence of chlorophyll molecules and the carotenoids allows the plant to absorb light, and wherever you have higher absorption of light, there you have higher rates of photosynthesis. What we are going to try and understand is how does this light energy drive the process of photosynthesis? Well, what happens here is that when you have a chlorophyll molecule, as depicted by this picture over here, this chlorophyll molecule is in the ground state. So it is in the unexcited state. When you have energy in the form of light hitting this chlorophyll molecule receives this extra energy and it gets excited. When it gets excited, it jumps to an excited state, and when it jumps to the excited state, it needs to release this excess energy so that it can come down to its ground state. And this is in fact the primary trigger for the process of photosynthesis. So when this chlorophyll molecule is in the excited state, for it to come down to its ground state, it passes off the energy in the form of electrons to a series of molecules, and these series of chlorophyll molecules will ultimately shuttle the electron to what we call as the reaction center, and in this reaction center the energy in the form of electrons will then be transferred along a series of other molecules to finally accomplish the process of photosynthesis. Now, the first thing we have to try to understand is that plants contain what we call as photosystems, and these photosystems are actually regions of the chloroplast where you have this movement of energy in the form of electrons. So they move within these photosystems, and their movement within these photosystems allows the plant to accomplish the various reactions that are very, very important for driving the process of photosynthesis. So we have in plants what we call as photosystem 1, and also we have what we call as a photosystem 2. And we will deal with these photosystems in much more detail in my later classes, but for now, what we only have to try and understand is that these two photosystems are driven by light of different energy. So one is driven by light in the wavelength of 700 nanometers, and the other one, which is photosystem 2, is driven by light in the region of 680 nanometers. When we try to understand the movement of electrons through the photosystems, what we have to first understand is that what happens in the photosystems is a process of what we call as charge separation. Now, what is charge separation? So example, you have two neutral spheres like this, and they have equal number of positive and equal number of negative charges. And what we know is that when we bring a charged rod near these spheres, then what will happen is that all of the negative charges will then move towards the charged rod, and all the positively charges will move away from the charged rod. When this happens, we have a condition of what is called as charge separation. Now, in the chloroplast, a similar situation also arises where the movement of electrons within the chloroplast brings about a condition where you have charge separation within the photosystem. Now, how does this happen? So what we have over here is you have light energy which is absorbed by the chlorophyll molecules, and this light energy is then passed on to the reaction center. And when this energy is passed on to the reaction center, the chlorophyll molecule over here gets excited, and when it gets excited, it jumps to an excited state. And for it to come down to its ground state, what it does is it passes off its electron, it passes on this electron to what we call as the primary electron acceptor, which is a quinone molecule. And when this electron is passed on to the primary electron acceptor, what we have is that the quinone molecule now receives an extra electron, while your chlorophyll molecule, which is present in the reaction center, is now in fact electron deficient. So when you talk in terms of charges, then the quinone molecule is negatively charged because it has received the extra electron, and your chlorophyll molecule at the reaction center now is positively charged because it has given off its electron. When such a condition arises, then we have a case of charge separation within the reaction center. And this charge separation is what actually triggers the successive steps in the light-dependent reaction of photosynthesis. Now, another thing that we have to try and understand is there are oxidation, reduction reactions that drive electron transfer within the process of photosynthesis. And before we get to trying to understand how does this happen, the first thing that we have to try and understand is oxidation normally refers to gaining of oxygen, or when a molecule loses a hydrogen, or when it loses one or more electrons. So any of these three conditions is a case of oxidation. And on the contrary, when a molecule loses, or when an atom loses an oxygen, or gains a hydrogen, or gains an electron, then that is a case of reduction. So what we have here is just an example where when you have your magnesium reacting with oxygen, and this is the overall reaction that you have, then if we break down this reaction, we find that magnesium, in this case, loses an electron, and therefore it becomes positively charged, and therefore we call this as an oxidation reaction. And oxygen, on the other hand, gains electrons, and therefore this is referred to as a reduction reaction. So what we find is that when you have a reducing agent, then this reducing agent will lose electrons. And when it loses electrons, therefore it itself becomes oxidized. So when X loses electrons over here, then X becomes oxidized, and Y, when it gains these electrons, then Y becomes reduced. So what happens is that your Y oxidizes X and your X reduces Y. So therefore X is your reducing agent and Y is your oxidizing agent. These reactions, where you have both oxidation and reduction, are what we term as redox reactions. And we can understand this better if we look at this following example. So in this following example, you have your stannous reacting with ferric to give you stannic ions and ferrous ions. Now in this case, what we find is stannous, your Sn+2, loses 2 electrons and therefore it reaches the Sn+4 oxidation state. So it is oxidized. And your ferric, it gains electrons and becomes ferrous, and therefore because it gains electrons, it is therefore reduced. And if we compare this reaction with the above reaction, the X and the Y type of reaction, then what you find over here is that your Sn+2 is the reducing agent and your Fe3+ or ferric is the oxidizing agent. Now further on, when we look at this, what we can tell is that this entire reaction can actually be broken down into half reactions, where one half reaction is an oxidation reaction and another reaction is the reduction reaction. And these two reactions in fact can be given what we call standard potential values, or what we term as electron volts. And what we have to try and understand is that whenever you have two half reactions in a redox reaction, then that half with the higher E0 value will gain electrons, and the half with the lower E0 value will lose electrons. So the one with the higher E0 value, that will undergo reduction, and the one with the lower standard potential value, that will undergo your oxidation. So what does it mean? It means that the higher the E0 value or the standard potential value, then the stronger is the tendency of that particular compound to gain electrons. If you compare these two values over here, what you find is that this particular reaction has the higher standard potential value, and the conversion of your stannous to stannic, this has the lower standard potential value. So therefore this one will gain electrons and be reduced, this reaction, and this reaction will lose electrons and be oxidized. And understanding the standard potential values is very critical in trying to understand how the process of electron transfer takes place during the process of photosynthesis. And what we're going to see is that these standard potential values are actually what determines the direction of electron transfer in the process of photosynthesis. And when we look at electron transfer, what we will see is that this reaction that you have over here, which is the reaction of the P680 in photosystem 2, this is a very strong oxidant, because when we will look later on, we will see that the standard potential values over here are very, very different from the standard potential values that we will find over here, which makes this part of the electron transfer to be very strong reductants, and this molecules, these compounds over here of the electron transfer, are very strong oxidants. So if I focus on this particular image that you see over here, you find that these are the standard potential values, where we have 0, and then you have increasing standard potential values towards this side, and then here you have decreasing standard potential values because these are on the negative side. And what you will find is that when your electron gets excited, what we will find is here this is a very strong oxidant because it has a high standard potential value. And when it has a high standard potential value, when it gains energy, then this jumps up to an excited state, and when it jumps up to an excited state, its standard potential value vastly decreases. And when it decreases, then it can pass off its electron from P680 excited condition. It can pass down its electron to the next electron acceptor, which is your phaophyton. And when you look at the position of phaophyton in terms of your standard potential values, you find that phaophyton has a higher standard potential value compared to the excited P680, which is around this region over here. And then phaophyton will then pass it down to plastoquinone A, and plastoquinone A also has a much higher standard potential value. So therefore, plastoquinone A will be readily reduced by receiving the electrons, and phaophyton will be readily oxidized because it will give off its electron. And this entire process, when you see at how these different compounds are aligned, you will find that in the transfer of electrons in the light dependent reaction of photosynthesis, or what we call the electron transfer chain, you find that there's always a movement of electrons from molecules that have a lower standard potential value to those that have a higher standard potential value. And this is what determines the direction of electron transfer, and the direction is therefore determined by the standard potential values. And you look, as you follow the path of the movement of electrons, it goes from plastoquinone A to plastoquinone B, enters the cytochrome B6F complex, and then goes to P700. When it reaches P700, there is another input of light energy, and this input of light energy further excites this P700 molecule, and when this is excited, then it reaches a standard potential value which is much lower compared to the successive molecules to which it passes on its electrons, and then it'll keep on passing down its electrons till it reaches the final electron acceptor, which is your NADPH. Now, the idea behind having this particular diagram is for us to try and understand that the direction of electron transfer in photosynthesis, and what we see here is what we call as the non-cyclic electron transport chain. The direction of this transfer of electrons is determined by the standard potential value of the different compounds that participate in the electron transport. And in this case, we refer to this as the non-cyclic electron transport or the non-cyclic electron transfer, because the electrons that originate from P680, these are transferred along this electron transport chain, and they finally end up in NADPH. So this particular movement or this particular transfer of electrons is unidirectional, and therefore it is called as the non-cyclic electron transport chain. Apart from the non-cyclic electron transfer chain, what we also have is another form of electron transport in your photosynthesis, and that is what we call as the cyclic electron transport. Now what happens is, we when we look at the non-cyclic electron transport, what we find is that the electrons move from P680 all the way through the different electron carriers depending upon their standard potential values, and finally end up in the final electron acceptor, which is NADPH. The thing we have to understand is NADPH, as we will see later on, is then utilized by the cell in what we call as a light independent reaction of photosynthesis. But there can be conditions where you do not have any free NADP plus to receive the electrons, so that NADP plus is converted to NADPH. So you may have a condition where you have electrons flowing all the way from P680 and landing up here in NADP plus, but then there are no NADP plus over here to accept the electrons that have come down all the way from P680 through PS2 and PS1 all the way down here, and the electrons have reached here. But when they reach over here, there is no NADP plus to receive the electrons. So what does the cell do? The process of electron transfer will continue. The process of the movement of electrons will continue, because as long as there is light, you will have excitation of the molecules, and when you have excitation of these molecules, you will have movement of the electrons, because the electron hole which is created over here, this electron hole gets filled up. So when they are filled up, then the entire process of charge separation takes place again, and the movement of electrons continue. But when they reach over here, electrons have nowhere to go. And when the electrons have nowhere to go, the plant cannot stop the process of electron transfer. So what it does is it shuttles back these electrons from your primary electron acceptor that you have over here. It shuttles these electrons back to your cytochrome P6X complex, and when they are shuttled back to your cytochrome complex, then these electrons will then move again through platocyanin to your photosystem, and this process will keep on happening. As electrons move from here, they come all the way down here and they reach over here, and when they have nowhere to go, then what will happen is that they will be reshuttled, and you will continually have a cycling of the electrons in photosystem 1. And this cycling will take place and continue to take place till you have NADP plus available to receive the electrons that arrive from photosystem 2 through photosystem 1. And because this movement of electrons happens in a cyclic fashion, therefore this is called as cyclic electron transport. And what we have to try to understand is that cyclic electron transport only involves photosystem 1, whereas your non-cyclic electron transport or your non-cyclic electron transfer involves both photosystem 1 and also both photosystem 2 and photosystem 1. And the other thing that we also have to understand is when you have cyclic transport, you do not have production of NADPH, but you only have production of ATP, which is produced by what we call as phosphorylation or photophosphorylation. But in your non-cyclic electron transport, you have both production of NADPH and also the production of ATP by photophosphorylation. So when we understand this entire process of the transfer of electrons, which is driven by light in the process of photosynthesis, or what we call as the light dependent reaction of photosynthesis, it then also allows us to understand how herbicides work when they are applied to unwanted weeds. And the two examples that I've given over here is one which is called DCMU and one which is called paraquat. And these two herbicides function by interfering with the electron transfer during the process of photosynthesis. So what DCMU does is, it does not allow electrons to move from plastoquinone A to plastoquinone B. So when electrons move all the way from P680 to plastoquinone A, and then they have to move to plastoquinone B, DCMU hijacks the electrons, and as a consequence you cannot have movement of electrons from plastoquinone A to plastoquinone B, and therefore it interferes with the production of ATP as well as with the production of NADPH. So you do not have ATP production, you do not have NADPH production. On the other hand, the other herbicide, which is called paraquat, this does not interfere with ATP production, but what it does is it interferes with the production of NADPH. So when you look at these two herbicides, though they function at different points of the electron transfer chain, they can interfere with the movement of electrons, and when they interfere with the movement of electrons, they stop the production of ATP or NADPH, or they can only stop with the production of NADPH, as in the case of paraquat, and therefore they are very, very effective herbicides because the plant requires NADPH and requires ATP to carry out the second part of photosynthesis. Now we've talked about the production of ATP, and we've talked about the production of NADPH, and we have tried to see how does this take place during the light dependent reaction of photosynthesis. The other very important reaction that we have in photosynthesis is the process of oxygen evolution. And this oxygen evolution takes place only in the photosystem too, and it takes place in what we call as the oxygen evolving complex, or what we call as the water oxidizing clock. And the function of this water oxidizing clock is very, very critical in determining the evolution of oxygen during the process of photosynthesis. And what we're going to do in this next slide is we're going to try and understand how does this process of the evolution of oxygen take place in the water oxidizing complex. So what we have seen earlier is that oxygen is produced as a result of the splitting of water. But how does that really happen? So what we have learned thus far is that your P680 is excited by light, and once it's excited by light, it gives you the excited P680 molecule, and this excited P680 molecule passes off its electron to the primary electron acceptor called pheophytin, and then pheophytin will then pass it to quinone, resulting in what we call as the charge separation, and then this will move on to the electron transport chain. Well, when that happens, what we have is a condition where you have an electron hole which is generated due to the loss of the electron. When this happens, what we find is that your manganese that you have in the water oxidizing complex, this manganese will simply pass off its electrons. So the manganese that you have over here, this manganese which is in the zero oxidation state, this manganese will simply pass off the electrons that it has to your P680, and when this is passed to your P680, this movement of electron takes place through a specific amino acid called tyrosine. So what happens is manganese, the ground state, gives off its electron to your P680 that you have over here. So P680, and it gives off this electron through the tyrosine amino acid that you have. And when it gives off its electron, manganese then gets oxidized to give you the manganese plus 1 oxidation state. Now when that happens, manganese plus 1 oxidation state can give off another electron again through tyrosine, because what happens is when P680 plus receives electron from manganese 0, that's the ground state of manganese, it receives the electron, this P680 is converted back and the electron is filled, so P680 becomes P680 plus becomes P680. Now when this P680 is there, it is ready to receive another photon of light. When it receives another photon of light, then the entire process continues again. So you have the movement again of the electron from P680 to pheophytin, and then from pheophytin it moves to the electron transport chain, and then what happens is we have another condition where P680 is electron deficient. When this happens, then your manganese, which is manganese plus 1, this will give off another electron through the same process, and when it gives off another electron through the same process, what will happen is that this one will give off another electron to P680, that's the electron, and then it will change to the next oxidation state, which is Mn plus 2. When you have Mn plus 2, then this electron from here will then fill up the electron hole, and then this will revert back to its ground state. Then again you have another photon of light, and this again will move through the entire process, and when you have the generation of P680 molecule again, then what happens is Mn gives off another electron, so Mn plus 2 becomes Mn plus 3, and then when that happens, what you have a condition is that this electron again moves from here through tyrosine, and when it moves through tyrosine it fills up the electron hole, and this P680 again gets filled up, and you have it ready to receive another photon of light. Now this continues 4 times. So when you have another electron from Mn3 plus is given again to P680 plus through tyrosine, then what we have is that manganese is converted from Mn0 to plus 4 oxidation state. Through these series of steps where it goes from Mn0 to Mn plus 1, Mn plus 1 to Mn plus 2, Mn plus 2 to Mn plus 3, Mn plus 3 to the final Mn plus 4. When it is in the Mn plus 4, the 4 oxidation state, then what happens is the water molecule that we have, this water molecule is split up, or this water molecule is oxidized, and as a consequence what you have is the electrons which are generated. These electrons are then taken up by manganese, and as a consequence when that happens, manganese plus 4 reverts back to manganese 0, and when it reverts back to manganese 0, then the entire process can take place again, and when manganese reaches its plus 4 oxidation state, then water again gets reduced, and when water gets oxidized, and when water gets oxidized, again it passes off its electrons to the manganese complex, and the entire cycle continues. And in this process you have generation of protons, and also very, very importantly, you have the generation of the oxygen, which is a result of the movement of your electrons to fill up the manganese complex that you have. So this, what we've seen, is what we call as the water splitting activity of the oxygen evolving complex, also what we call the water oxidizing clock, and this is the mechanism by which the process of electron transfer during the light dependent reaction of photosynthesis drives the splitting up of water to produce oxygen, or rather drives the oxidation of water to give you oxygen. If you liked this video, then do not forget to like and comment. Subscribe to my channel to watch upcoming videos.